Hi, Krishna.
அங்கே ஏதோ ஃபோன் இருக்க மாட்டேன் எடுத்து வச்சு சார் மீட்டிங்கில் இருக்க சொல்லி
सर मेनाथन सर
மாறிக்கொள்ளணும்
मेनाथन सर
மாறிக்கொள்ளணும்
இங்க வாங்க
மூஞ்சி தெரியாது இவனுக்கு
ஸ்டார்ட் பண்ணுங்க ஜெய்சி சார் ஜெய்சி ஸ்டார்ட் மாஸ்டர் மியூசிக்
A warm good evening to one and all present in this wonderful evening. I am Dr. M. Jayaprakash, Assistant Professor from Department of Pharmaceutical Analysis, JSS College of Pharmacy, Uti. With blessings of His Holiness Swamiji, we would like to start this webinar. As Organizing Secretary, I extend my warm welcome to on behalf of JSS Academy of Higher Education and Research. I submit my sincere thanks for your valuable presence for this international webinar on impurity profiling of pharmaceuticals. Organized by Department of Pharmaceutical Analysis in association with Institution Innovation Cell and IIIC JSS College of Pharmacy, Uti. I hope this webinar will enlighten the participants' mind in the impurity profiling research area, which is considered as a very important topic and need for the day. With this few words, I request our beloved principal to deliver welcome address and introductory remarks. Please, sir.
I submit my humble and sincere pronounce to the lotus feet of Jagadguru Sri Sri Desindra Maharaj Swamiji of Suturmat and Chancellor of JSS Academy of Higher Education and Research, Mysuru, and seek his blessings for the success of this international webinar. Despite the COVID crisis pose a significant challenge and the aftermath impact is projected to be worse than COVID, we at JSS are continuously encouraged and mentored to move forward with a resilient notion that learning never stops at JSS. I take this opportunity to thank the senior leadership for their extended belief and positive leads. Now, should I say good morning, good afternoon, or good evening? Well, it appears the sun never sets in virtual kingdom. I, Dr. S.P. Danpal, Principal of JSS College of Pharmacy, Uti, extremely happy to welcome you all, resource persons, panelists, and participants across the nation and globe. I recollect that in 1990s, I once taught him pharmacognosy subject without even realizing that I would be listening his expert lecture. But as a teacher, it's always a pride to cherish the success of their students. I'm extremely happy to welcome one of my beloved students, proud alumnus of JSS College of Pharmacy, Uti, as a resource person for the expert lecture, Mr. Ravi, co-founder, president, and CEO of Citrogen Pharmaceuticals, California, United States of America. Welcome back, Ravi, and my best wishes for today and ever. I'm equally delighted to welcome one of the emerging experts in the field of pharmaceutical analysis, Dr. Saravanan from Spinco Biotech, Chennai. The portfolio of Dr. Saravanan clearly demonstrates that he's vibrant and passionate towards his fields and deserve the credentials. Welcome, Dr. Saravanan, and my best wishes for you as well. So this international webinar is being hosted by the JSS College of Pharmacy, Uti, initiated by the Department of Pharmaceutical Analysis, jointly with the Institutional Innovation Council and Industry Institution Interaction Cell. I collectively congratulate Dr. N. Krishnaveni, head of the Department of Pharmaceutical Analysis, and her team members comprising Dr. S. N. Mayanathan, Dr. J.S.K. Naharajan, Dr. Jayaprakash, and Dr. Babu for the right topic with the right experts as panelists and right time. All your efforts are greatly appreciated and best wishes to you all. Last but not the least, dear participants, evidently, we millennials are blessed with a technology advancement. Fortunately, today we reach your doorsteps along with eminent experts in the fields to nurture and groom young minds as well as keep your learning consistently constructive. Please use this platform and opportunity and benefit. Wish you all the best and all success. Thank you.
Thank you, sir, for your wonderful welcome address and introductory remark. Now, I'd like to request Dr. N. Krishnaveni, Professor and Head, Department of Pharmaceutical Analysis, to introduce the first speaker.
Thank you, sir. I'm extremely delighted to introduce today's first speaker, Dr. S. Saravanan, the Principal Investigator at Spinco Customer Service Center, Chennai, Tamil Nadu. He has been practicing analytical science over a period of 16 years at various multinational Indian companies, and he had handled the sophisticated instruments like GCMS, LCMS, ICP-MS, NMR, and many others. He was awarded with PhD from Bharatiyar University for the topic entitled Impurity Characterization of Type 2 Kinase Inhibitors. Prior to his recent assignment, he has been associated with Piramal Healthcare Limited, Signrise, and he was also in academia.
Next. Next.
He has also served academia as a lecturer at AM Jain College, Chennai. And he had presented his research work in various international conferences held at Canada and Japan. And he had delivered lectures on chromatography, mass spectrometry, and other sophisticated instrumentation techniques to various organizational gatherings, especially at Madras University, Manoanmaniyam Sundarnar University, Annamalai University, Periyar University, and also Tamil Nadu Pharmaceutical Sciences Welfare Trust. With this small introductory remark, now I request Dr. Saravanan to give his lecture. Thank you.
Thank you, ma'am. Over to Saravanan, sir.
Thank you for the dais members. I'm honored to share my experience and exposure in this scientific webinar society. Let me share today's presentation screen. Before starting, I would like to confirm two things. One is, hope my voice is audible and my screen was shared.
Yes, sir. It's very clear. You can proceed, sir.
Thank you. Thanks for the wonderful introduction, and I am very much honored to share my experience and exposure to the fellow scientists of tomorrow, students from JSS College of Education, and of course the faculty members who are having more knowledgeable than all of us, because day in and day out you are doing more things in the academia. Of course, all the more research things are also happening in the JSS University. Now, let us move towards the topic. So today's topic is impurity profiling in pharmaceuticals. As such, the topic is very vast. Impurity, is it a major concern actually? To say, is it a major concern? Yes, of course. Even today, the stringent impurity, what the pharmaceutical peoples are looking for is nitrosamines. So even today, one or the other is coming into picture as impurity, not as ingredient, having its own stringent effect in the biological stream. In that case, being a pharma graduates, being a chemistry or pharma background students and the faculty members, we should know what is this impurity, how this can be profiled with the sophisticated instruments or the analytical techniques what we have, for the welfare and the betterment of the process, which involves the new drug discovery as well as drug processing. Not only that, India is, you know very well that India is a very good generic market, and in generics, this plays a vital role, and it needs a match to the innovator always. With this introduction, let me move into the slides. So today our discussions are mainly towards these 5 things. Impurity. What do you mean by impurity? What the definitions and all about is telling for you. Its origin, the key regulations. I'm not going to go much into the regulations because regulation session itself is a lengthier one. So I just would like to touch the key regulations, solutions by LCMS. Though I am stressing LCMS, I'll just cover the few analytical or sophisticated instruments which plays a vital role in this impurity identification. Not only that, if you say impurity profiling, it also involves the isolation of the same with proper characterization so that it attains its completion as a known impurity, which is in the manufacturer control to have a better product for the society. So this is our discussions, and feel free to ask any questions. You can type it in the box, and I'm happy to take it up and discuss at the end of the session. So let us move on. Before going towards the impurity and its definition, we must know what is the pharma workflow actually. What is the pharma? Is it changed, or what? To what extent it has been changed? So there are 3 major stages in any pharma workflow or pharma process. One is the R&D stage, the other one is the pilot study stage, the other one is the plant stage. So mostly in India, we are focusing on the production plant, but still there are notable R&D and pilot stage, pilot study players are still there in India, of course. So their strategy will be synthesis of the drug molecules and analysis for development of drug molecules in different entities. So this forms the basis where they will be keep on doing much docking studies, and they'll be concentrating more towards the product based. But once it comes into the finalized and the medicinal with the medicinal value, then they will start to have the investigation for all. Earlier, R&D means it has to deliver a good product to the society. That's all. Now, the good product has its own definition to carry out. What do you mean by good product? Is it the one to have an assay of 99% to cater its intensive application or intensive action as a drug? Not only that, the safety of the drug, the way in which it is getting manufactured, the way in which it is getting scaled up, also focused now. That's why now the regulations are moving towards R&D to scratch the base of the production of the plant. Next, it comes to the pilot scale study where the so-called or the obtained drug products and obtained drug ingredient, both cases. It is happening in both cases, the ingredient as well as the drug product. To scalability, to what extent it can be scalable, its reaction values, its intermediate stages, where the pressure, environmental safety will be taken care of, to have a hassle-free timed operation to focus on the delivery of a particular entity. So here the focus is not on the safety much, yes, here on the environmental focus, and also to have the product in a particular specific timeline. To have that, a BMR and other things will be in a mock trial. You say it's a pilot plant. Then it comes to the production plant. So it has the mass scale production where all the trials are finalized. The drug has its own, drug was accepted internally and externally in local market and the international market, that it went on to the production scale with different scales, scalable size. So the quality control of final drug and formulation plays a vital role. I'm again asking, I'm audible, right? Hello?
You are audible, sir. You can proceed, sir.
Yeah, fine. So these are the 3 major process So in this process, now impurity, earlier it was impurity was concentrated only in the production and the plant level. Now the impurities are started to get monitored right from the basic research level itself. So you have to provide the possible probable impurities or carryovers from your process or the interactions with which it can takes place. So it started from the R&D place itself. So by definition, impurity, any component of the new drug substance, which is not the chemical entity defined as a drug substance, or any component of the drug product, which is not the chemical entity defined as the drug substance, or the excipients in the drug product. So this is ICH1, and it has a major classification from 1 to 3. So this is the impurity, because I may say one, you may say one, and people may have their own imaginations on impurity. So there should be a definition, and there should be a perfect definition in both the drug substance as well as the drug product. In both cases, if you see, it clearly states that any entity apart from the chemical structure related to the drug or the drug substance or the ingredients what you add. So there is a clear differentiation between the ingredients what you add and the drug and an impurity. This is very important, because we can say that a pinch of salt in the 1 ton of chips for the taste. Do you think that salt is an impurity or an ingredient? Then in that case, if you say it will become an ingredient. So you should be well enough, or you should be in a position, or the definition should be defined in such a way that it should be differentiating the ingredient, whereas it should cover the rest of all. That's why any component of new drug substance, which is not a chemical entity, which means you have to define the drug substance or the drug product properly with its formulation. Apart from that, whatever it appears, it may be an intermediate, it may be a solvent base, it may be a degradant process, it may be an overcooking impurity in the production scheme, whatever it may be. If it is not the chemical entity defined as a drug substance or as a drug product, then it is coined as an impurity. This is what regulatory says. Fine. Now, as we all know, pharma is mainly dependent on the base of chemical reactions, and a lot of active reactions has to be taken care of in a safe manner to have a proper output. Based on this, all the chemicals can be grouped together. If I say chemicals, then it can be organic as well as some physical modification, organometallics may also come into picture, based on which the impurities was also considered for its own allocations as broad classification as class 1, class 2, and class 3 impurities. So this is the class 1 impurities. It is nothing but organic impurities, inorganic impurities, residual solvents, polymorphic forms, enantiomeric impurities. Very closely related to the process, very closely related to the solvents, or very closely related to the compound even. It may be having a different polymorphic form also. That is also now categorized as a class 1 impurity. And definitely enantiomers, it is a major role to play. It is very much equivalent to your drug, but it is not your drug. So in that case, enantiomeric, chiral impurity also plays a vital role, and it comes under class 1. We all know that. So what do you mean by class 2 impurities? These are also organic impurities, but can raise during the drug manufacturing process or process development, what we used to call as a PD, process development job, day in and day out in the case of generic market. At the same time, they can also be identified. They can also be identified as byproducts, intermediates, degradation products, mainly degradation products. This plays a vital role. The reagents, the ligands, the catalysts. The catalysts also coming into class 2, because the catalytic removal or the catalytic trace will also has its own effect. Then the class 3 impurity. Class 3 impurities is quite inorganic. That's why I said organometallic can also come into picture, but it is in the case of class 2 if you define the metal as organometallic. But if you define the metal as a metal, then it comes to class 3, that is inorganic impurities. Can result from the manufacturing process anywhere. It may be a leaching thing from a leaching element from the plant containers. That is also possible. It may be a common reagents which is not very specific to this drug products alone. It is a common one which was quite common usage for many other drug product and drug ingredients that will also come into class 3 kind of impurities. Heavy metals, of course, many heavy metals are there. Even if you are using a high inorganic content of water, few cases also there is a heavy metal contamination of water. If you are using that water for the process, there is a chance that your product may also contain the heavy metals like lead, arsenic, etc. Inorganic salts is, of course, if your product itself is a salt, it can be a potassium salt, it can be a sodium salt, but that will not come under the impurity status. If you say that your product is a potassium salt, but if you are having something as a say sodium, or if you have something as other electropositive elements attached to your product, and your assay may pass, but come together when you do an analysis with sophisticated instruments, it may fail with different salts or different inorganic metals present, metallic presence. And other materials, filter acids, or even the charcoals, the drying agents, the silica gels, which are carryover hand pickable. That is also coming under the class 3 impurity. So these are the main impurity categorization. This was categorized based on the drug ingredient manufacturing and the drug product and the placebo, whatever the placebo manufacturing. All together, it put together to have a broad classification into 3 major categories. So these are the 3 major categories, and we should understand 3 major categories so that whenever you come up with a process, whenever you interact with a drug, or you wanted to go for a publications where your impurity is an organometallic, then it has to be coined as class 2. So if it is a metal, then it has to be as a total metallic content in class 3. So this plays a vital role, and accordingly the limits will also change. Now we know what's impurity. So what is the definition for impurity? How the impurity can come into your product? What are the 3 broad classification based on the process, based on the solvents what you use? Fine. Now, fine. Based on this, how my analytical technique will help me in identifying or in categorizing these impurities or quantifying. Both characterization and quantification has to happen, because identification is the first step. But pharma doesn't stop with identification. You have to characterize. For characterization, you need isolation. So isolation is also a key. So the impurities, as I classified as inorganic, organic, and volatile, there are different sophisticated techniques available. ICP-MS is the most important technique as far as the inorganics are concerned. That's why even now the pharmacopoeias are asking to remove, almost now it is removed, I think, the ash content, which is the conventional analysis to get the total inorganic content, instead of which they are asking to perform the sophisticated ICP-MS, the inductively coupled plasma mass spectrometry, which is one of the most interesting and important sophisticated analytical tool to determine and quantify both qualitative as well as quantitative determination of inorganic elements in the elemental scale. If you are having some organometallic methyl mercury kind of compounds where it is a poisonous one, at the same time it has to be identified as a methyl mercury as such. In that case, ICP-MS may not give you a proper result. In that case, you have to go for an AAS. So AAS will give you a proper result, or you have to go with a hybrid MS where the identification not only goes with the identification of the MS, but the specificity of the analysis also remains the other chromatographic techniques. Say example, LC-ICP-MS. In that case, mercury, methyl mercuries can be separated and can be sent into the mass spectrometer, where that ICP-MS is a hybrid one. In that case, ion chromatography will also come into picture in the case of LC. Though we are having some topological methodology is also available, ICP-MS is quite common in the case of specific methyl mercury analysis. Otherwise, straightforward, they will go for an AAS. So you have to choose the proper analytical technology to cater these three classification of the impurities. Of course, by organic, once I say organic, then if it has a chromophore, then we will jump and do the analysis directly with an LC UV or a PDA detector, where almost all chromophoric counterpart will come into picture. But what I will do with placebos, most of the placebos are sugars, polysaccharides. In that case, their chromophoric activities won't be there. In such case, I have to go for a bulk property detector, say for example, an LC coupled with an RI or an LC coupled with an ELSD. LC coupled with an ELSD is a powerful tool than the LC coupled with an RI. Why? Because you can perform an LC ELSD gradient combination, which is not possible with LC RI gradient. RI can be run only with an isocratic mode. So the methodology what you choose also plays a vital role in determining or quantifying the impurity in a proper manner. Nowadays, more sophisticated instruments are available. Earlier, just 15 years before, if we have a HPLC, we used to say that we are one of the institutions to have the most sophisticated instrument. But now HPLC is becoming a household instrument for almost all academic as well as pharma firms. Almost all the academic as well as pharma firms will have HPLC. HPLC is nowadays not called as a sophisticated instrument. If you have an MS, if you have an ICP-MS, if you have an NMR, then there is a chance. Now the sophistication moved itself into another stage. The CAD is there, and GC is also a vital role. Why I am stressing more towards the LC and LCMS? Because all our pharma products, or pharma ingredients, or pharma drug products are aqueous-based formulations or suspensions, because that has to be given to the living organisms, especially humans or the other living organisms. So almost all the living creatures in the earth, especially the humans, almost or with almost more than 80% of water content. Our metabolism is mainly a water-based metabolism only. So the drug what you give, or the drug product what you give, should also be an aqueous-based formulations, the aqueous intake, the aqueous-based. So once I said aqueous-based, then it is the suitable analytical technique will be of a chromatography for separation, which includes a liquid phase, which means a liquid chromatography will come in picture. Doesn't mean that gas chromatography will not at all be useful. That doesn't mean gas chromatography is required. Is must, because the process of different drugs requires organic solvent washes, organic cleanups to quantify the organic, to qualitatively determine the purity of the organic solvents. You need GC. Even in the case of GC and GCMS, impurity characterization and impurity profiling is must, and it is a bit simpler in the case of GC and GCMS also. Why? Because you are having an aid of NIST library in the case of GCMS analysis, which is not there in the case of LCMS analysis. Nevertheless, whether it is GC or LC, the ultimate end up for any pharma product will be a chromatographic instrument. It can be a GC based or it can be an LC based. And then for identification and characterization, definitely people has to come for the respective MS together, so that the GCMS and LCMS both will come into picture. So have we spoken about this impurity for the past, just before of 20 years? No, the conversations for the impurities are not that much actually. After the effect of impurity in different stages, in different levels, it is not only the purity of the product which decides the efficacy of the drug, but also the impurity which governs the overall health of the subject or the human to which the drug was given, for which impurity profiling is important. So what do you mean by then impurity profiling? Now we know what is impurity. Now we know what are the different classes of impurity. Now we are having different analytical methodology, how to determine and distinguish them into different categories and characterize them and quantitate them. Impurity profiling means we need to have the profile. So what my drug is going to provide in all course of the metabolism internally. So that is one part which is going for the bio. But impurity profiling lies with an external thing. So what are the trace level constituents carried from the process? What are the trace level constituents which can build in its life, in its shelf life of a product? What are the constituents which can be created by the drug with its interaction to the environment? It may be the spotting of the tablet seals. It may be a polymer which holds the drug as a suspension. All these things will be coming into picture. So each one will have its own name. Now they are further refined. This impurity profiling is getting refined to leachables and extractables. Actually, that will also come under this impurity profiling only in the broad classification. Now that is also getting refined with respect to the polymer, with respect to the packing, with respect to the interactions of this drug and drug product with the environmental containers, environmental suspension solutions. So that is different. So that is also coming into picture. Before going to that level, we should understand few analytical technology also, for which I will be explaining you with few examples of MS-based experiments. Well, I'm not going to touch with GCMS-based experiments or GC-based experiments, as they are much simpler actually. When you compare a GCMS and an LCMS, or you can compare an LC analysis with a GC analysis for this impurity profiling, the GC experiments and GC-based experiments are a bit simpler because everything is volatile there. The carrier gas is volatile, that it involves a lot of temperature. So thermal stability of the product and the impurity is also certified. That's why we are proceeding towards the GC or GCMS. In the case of LCMS, it's an aqueous based. And almost most of the formulations of the drug products and the drugs are very much aqueous based. So I need to go a polar solvent for the elutions, polar organic solvents for the elution chromatography. So pH comes into picture. More and more of polar functionalities will be coming into picture. So everything, the stationary phase, the mobile phase, and everything plays a vital role in this case of impurity profiling as far as the LC and the LCMS is concerned. I'm staying in this slide for a longer time to stress that the different technologies and the difficulties what people are facing and how they are coming out. Especially if it is a liquid chromatography, then we are having a well-known related substance method. We all know very well. All the pharmacopoeias will be guiding us, will be the Bible for the pharma people to provide the impurity profiling. Yes, of course. Before which they have to do a lot of exercise. Have to submit the data in drug master file, DMFs and other things. And based on that, only this pharmacopoeia and other things will be derived and given as a Bible. How they are doing that, this impurity profiling, in the case of related substances. In the case of LC-UV or LC-PDA, it is only the excess concentration of drug what you inject, say 5 times, say 10 times, depending upon the level, depending on the intake level. We used to say the intake level. It may be for the process or it may be for the function. In both cases, we have to decide, so say 5x time for impurity, so that the lower level of that impurity will be seen in the chromatographic session itself or chromatographic elution itself. In that case, excess concentration of API will be injected in the chromatographic stream to see the low level concentration of unknown entities. And then they will characterize one after the other, and to put together, they'll come up with, say, okay, no matter whatever the process you follow, no matter whatever the process the X follows, the Y follows, the most probable and possible impurities for this product A has 7 things, 7 impurities that will be displayed. This is called the impurity profile. Fine. If some other new entities are found in, then you have to go to your retro analytical methodology to the process, the check, the modifications, what you have performed in your analysis, the changes what you have performed for the particular batch in your BMR, the batch monitoring record, is a vital role for this impurity profile. Fine. Now let me go towards the few examples. In the case of mass spectrometry, that is LC based, the LCMS and MSMS mass spectrometry. So in the case of LC MSMS, we are having one Pandora's box, nothing but the interface, because in the case of GCMS, the Pandora box is quite closed, because everywhere it is volatile. Whatever you inject, it goes into the column, and whatever the impurity profiling you want to search, whatever the as such you will get in the case of GCMS integrated detector. If you it can be correlated in a single stretch because it's a radical based hard ionization technique. So you can directly relate it to the NIST based library, which works under 70 electron volt. Fine, that is concluded. And much evident also in that case. Whereas in the case of LC based, I cannot apply my 70 electron volt for an LC-MSMS or LCMS analysis, because LC based molecules are polar in nature. So if I'm going to supply that much energy to the compound or the impurity I'm going to see as a profile, then it shatters and pieces to see only few functionalities, maybe CO2s, maybe small fragments. That is not my interest. I want to see the molecule intact as a molecular ion, and then I wanted to see the building block of the molecule as a fragmentation, so that I can have a meaningful fragmentation pathway for the mathematical M by Z values. The same can be compiled and correlated for the impurities to have a better understanding for the impurity profiling. This is what my intention in the case of LC-MSMS approach, for which the ionization value should be so low. That's why it is called as soft ionization. So once I say soft ionization technique, it's only maximum of 5 kilovolts. Somebody is giving something as 5.5. Nevertheless, almost all the pharma molecules, what we are dealing now, which is in the molecular weight range of 150 daltons to somewhere around 900 daltons. I'm not speaking about the short peptides. It's only the monomers, which covers the entire range of this molecular size, needs only this much energy. If you are doing a bit more energy also, it is as equal to that of overcooking and making thing a complex one in the spectral analysis or the interpretation one. Fine, that is not my motto in the case of impurity profiling. I want to be a bit clear with clarity. Let me proceed with few examples. So before which, one regulations is, of course, is very important for the dosage and the threshold limits are concerned. You can see here the maximum daily dosage is less than 2 grams per day. This is for drugs which are of say cardiac based, which are of say hypertension breaks. Almost all these drugs are less than 2 grams per day. If at all the patients are very much severe, higher dosage levels will be prescribed, very specific by the practitioner, that is doctors. Otherwise, it will be always less than 2 grams per day. Few drugs, of course, yes, it is greater than 2 grams per day. Say, for example, even now nutraceuticals are coming into picture, but though nutraceutical contents are not to this much, few ingredients are added in such a high concentration that body is taking to that level and it is getting assimilated to that level. For that, if it is greater than 2 grams per day, which is quite very relaxed spec in that case, then it is less of 0.05% as identification threshold and 0.05% as a qualification threshold. So again, reporting and the drug products, it becomes stringent. If it is less than 1 gram as a drug product, then it will become 0.1. So the quantification or the identification threshold is very, very stringent in the case of drug products, very, very stringent. And its interaction is also very much, many pharma they will prefer to perform the assay rather than the assay they want to perform the as API. But for drug product, they are not that much reluctant to do the analysis for RS. Reason definitely they will find a few things as 0.1 or greater than 0.1. In that case, actually it is not hazardous. Actually, this is because of the small interactions which even the sample preparations can also give. Even the regulatory allows that. That's not a problem. So, in broad classification, in combinations, you can have the excipient interactions, the drug products, the different impurity type, and the impurity source. I have discussed much in detail on this slide. And based on the drug product, it comes an add-on to another two circumstances. Otherwise, everything looks same only. It's a replica for what you have as a drug substance. The same will hold for the drug product. Same is only, except the excipient which was added in the drug product. So now, as I inform you, I will just go through a few examples of mass spec because the time is not that high. I have to concise the exercises, the experiments, and a bit interesting too. So mass, we all have different spectrum when you give that. As it was connected, it's a sophisticated detector which connected to the chromatographic instruments. It can be a GC, then it is GCMS. It can be an LC. It can be then it is an LCMS. In all the case, what you are going to do is to see it's to identify the mass, where you will get a spectrum. What do you mean by spectrum? Spectrum is nothing but mathematical. A spectrum is nothing but a graph with mathematical values, meaningful mathematical values. That is what important. That is what required for the interpretation. Because even the simple solvents or simple air can give you some spectrum, but it is not of interpretation sake or repeatable sake. So it's a time-based technique. A simple analogy, what I'm going to do in a mass spectrometer, I'm just bombarding. Now you know that this is of a pot shape. You're colliding, you're breaking the things, and then you are refabricating to the structure. This is what the structural elucidation. This is how the structural elucidation was performed. You don't know that that is a pot, but you are bombarding with a beam of electrons to the respective fragments. Patiently have to rearrange it. It may be through software, it may be through your knowledge, it may be through your interpretation with respect to the free radicals and the connectivity to that and to the mass spectrometer, whatever it is, you are regaining the pot. Then you are seeing that and saying that it's a pot. That's how the structure was assigned. It's a simple analogy. So this is how now you know very well about the molecular ion peak, the base peak, the fragmentation peak. So the different fragments. Why I'm telling these things what it is related to the impurity profiling, because these are the areas where people go mislead with respect to the base peak and the molecular ion peak and impurity. Because in the case of impurity profiling, it's again you need a lot of evidences to say that it is a impurity and it is repetitive for the process and it is appear. You need data. You need to provide enough data to coin an impurity as an impurity. And to differentiate that as a known impurity and an unknown impurity, you still you need furthermore data. To have such authentic data, you need both otherwise you know the process. If you know the process, yes, of course, LC or GC retention times with the standard spiking, but they also the profiling should match. So it is not only the drug which decides the efficacy. Now even the profiling matters for almost all the drug product. So the basic setup you can see here. As far as an LCMS is concerned, ionization has to happen in the atmospheric pressure. Analyzer and reduction. both in the case of GCMS and the LCMS, it is perfectly vacuum. The beauty is, as far as the mass is concerned, for the small molecules, to see them intact as a single molecule, you need the molecule to get ionized. Ions are short-lived species. It cannot live in normal atmospheric pressure. I'm speaking, you are listening because my voice is getting telecasted through this web platform. That's why all of you are listening. But the same thing is not possible if these things are not there. Only you will feel, only if I switch off the mic, you will not listen anything. Though I dissipate energy, the energy may not reach even, it may not even cross my compound wall. So the energy is getting dissipated. Whereas ions are such a short-lived species, it can live only 10 to the power of minus 15 seconds, which means as and when they generate, it dies. To preserve the ion in its ionization stage, and it has to enter into the free path, that is the mean free path, you need a perfect instrumentation picture. And almost all the people are giving the perfect instrumentation picture to cater this technology. That's why that impurity profiling has grown this much. So majorly, these are the major three things. One is the ions get generated and get filtered in the analyzers. It can be a quadrupole, it can be a time of flight, it can be a TOF, it can be a TOF, the time of flight, it can be a trap. Whatever it can be, it has to filter. That is the job. That is the analyzer job. Then, though mass spectrometer itself is a detector, it also requires a detector, the EMT or the PMT, the multiplier tubes, which amplifies the incoming signal into an avalanche of signals to get readable by as an electronic signal in relation to the concentration of the incoming ions. That's why we are doing quantification in these ions. So this is what a mass spectrometer does. Fine. So I don't want to go much detail on the instrumentation type, but we need to know few things together. So how much a compound is present in a matrix is of course a quantification thing, which you need high sensitivity. Provides information also about the structure. That is also important actually in the case of mass determination. Though nowadays, simple quantification is not enough. You need to prove the quantification with multiple data in a same analysis, single run, single analysis. Where the parent frag or the parent daughter plays a vital role. Now the intensity of these ions also now coming into picture as ion ratios. Earlier we used to perform the analysis as one-to-one analysis, that is as an aqueous based analysis. Then later we used to have a relative analysis with an isotope or an internal standard method. Now it becomes quite complicated, that now even much more complicated with the drug product, where the interactions and now the intensity of the parent and the daughter in particular collision energy or particular environment matters, which again helps us to eradicate the false positive results. That is also we should know whenever the person is very much intended for an impurity profiling kind of analysis. So this is what an LCMS and LCMS. If it is a PDA, you will get an additional spectrum, whereas in the case of MS chromatograph, you can get an MS as well as the MS spectrum. There is an MSMS. Why I'm differentiating this as an LC? MSMS means in the case of GCMS, as when you eject, when the electrons are ejected, the parent molecule, the parent ion, appears along with its fragments in a single shot. That's why there is an ambiguity to call which one is parent. With an intensity, you call someone as a base peak, someone as a molecular ion peak, which if it comes first. Here in the case of LCMS, the same holds good, but there is one more terminology, MSMS. So in LCMS, you will get only the parent ion because it's a soft ionization. You have to do one more exercise of fragmentation, which means you need another analyzer setup to have an LC-MSMS fragmentation profile, which is very much mandatory if you go for a characterization or profiling exercise. Because in the case simply LC-MS, you will get only a mere mathematical number. Even in the case of GCMS, you will get the number, but the number will be followed by its fragments, which becomes the fingerprint of that entity, which can be again related to the library. Whereas in the case of LCMS, you will get only the mathematical value as a molecular ion or a representative ion of the drug, which where you need to perform one more operation of MSMS of this specified or obtained mathematical MZ value to get its fragments, which becomes the fingerprint of your entity. So there is a difference while dealing with these two techniques as GCMS and LC-MSMS. So this is an example, impurity characterization in a famous drug valsartan. Now valsartan is under very much scrutinization for this nitrosamine impurities. We all know it is a very good antihypertensive drug and very much supportive for heart attack cases where it immediately suppresses the BP in a very good manner. Of course, there are other drugs that are also available, but this is a well-proved and old drug and almost marketed by more than 30 countries. So this is an impurity profile by LC. You can see here, this is an impurity profile by LC. I'm just made it a bit more zoomed to show you the different impurities, what you see here. And label it with respect to the retention time as 1, 2, 3, 4, and the one which is appearing after the big main peak, that is the valsartan peak, as impurity 5. You can see here then. So these are the respective retention times. This is how the LC gives, and the concentration from LCs are there. And you can see here that impurities are not that great, but still alarming. So this not 1, not 2 is not that much great, but not 3, not 4 is alarming because you need to have a not 5%. If it crosses a not 5%, then it will become alarming. So people, as when you see that, will go through this point not 3, not 4. And impurity 3 is it's an USP kind of impurity. No matter what process you follow, it will be there. So it's a known entity. So this is the zoomed analogy, and this is the kind of spectrum which will be given. I'm showing this as an example because interpretation is the key in impurity profiling. No matter, it can be GCMS, it can be LCMS, even ICPMS. Even in the case of ICPMS, though we can say that it's only 108 elements in the periodic table, and only it is going to cater only the electropositive elements, so it comes hardly within 60. But in reality, and getting the data and transferring that for a perfect impurity profiling for an in the inorganic aspect, especially if it has a salt formulation as potassium salt or sodium salt, in that case it will be very tedious, but possible. So here, the trace level of different ions which is appearing. So from these ions only you have to extract. That is important. The case of extraction is important. So if you extract, this is the information which you got. So this is what instrument gives, and this is what the analyst gives. This is the difference between the profiling person, impurity profiling person, and the instrumentation handling person. So whoever be the instrument handler should know what is the objective of the analysis, so that he can become an analyst interpreter rather than an instrument handler, which is very much important. Otherwise, no, these are the basic information which mislead to a larger extent in the case of impurity profiling. And this is an MSMS spectrum for an impurity where we did more exercises, compiled the data to arrive at the spectrum's values. And then there is a comparison. And this is how comparison will be. This slide is important. You can see here. I know the drug valsartan, its molecular mass, I know that. Fine. Though I know the mass, I know the structure. Now I'm having its building blocks as ions or fragments from my MSMS. So I can have a proposed fragmentation pathway in a similar way. I don't know the structure of the unknown impurity 5, but I'm having the fragments of that impurity, which is having a common correlation with that of the main compound valsartan. So in a manner which I have used for valsartan fragmentation pathway, I will use a similar one for the impurity 5. And I propose a fragmentation pathway like this. the comparison will remain like this. So the mass value 436, 450, 235, 249. Everywhere, if you see, there is a difference of 15 units, the presence of methyl group. The 15 units, the methyl group can be present in the biphenyl ring, or it can be present in the tetrazole ring of the compound. It can be present as a substitution, external, exocyclic substitution in the biphenyls also. There is a possibility. But the mass fragments are not supporting to that. The mass fragments are supporting to have the methyl substitution in the tetrazole ring. And of course, this is a preliminary first step of identification or profiling that impurity to short down your predictable fractions. So these are all predictable functions. It can be a GCMS, it can be an LCMS, it can be a different hybrid technology. No matter whatsoever, it's only a predictable functions. At the end only, you will be having predictable possibilities. Then you have to isolate and confirm through NMR, which is the ultimate. And you should have that impurity in hand to authenticate. It can be a chiral enantiomer, it can be a normal diastereomer, it can be a process-related isomer, whatever it may be. So another characterization, I'll just rush through. So MSMS beyond NMR data. In some cases, to emphasize this MSMS when compared to NMR, NMR is the ultimate goal for end result. But for impurity profiling case, if you want to compare that with the case what we did it for the main compound in MS, it's not possible because NMR requires pure fractions to have its own signals. Otherwise, it will be a clumsy one and identification is not that much easy. So this is an impurity where 0.7% it was isolated, and this is the main compound, and this is the impurity 1, and you have the comparison here also. The unit is 15, but here the mass value is big. The main compound is this one, 606 Dalton, and the impurity possibilities are this. So these are the possibilities. It can be as a methoxy. The methyl can be as methoxy. The methyl can be substitution in the alicyclics. It can be a substitution in the aromatic. It can be a substitution before and after the sulfur group. So in either case, it is possible. If you compare that with the NMR, then the possibility of methyl identification is not that easy. Everywhere it remains same because in the aliphatic region, we are having too much of aliphatic chains. And differentiating that with respect to the intensity is not that easy for a molecule crossing 500 Dalton value, as it has more number of hydrogens. Of course, in carbon NMR, there will be some differentiation, but it is also not that much marked. And this is a comparison of the different collision energy. This technique is called as the NMS to the power of 2 technique in the triple quadrupole analyzer instrument. Of course, you can apply the same thing in the case of trap system with the NMS to the power of 2 technique. In the case of TOF, it is difficult because TOF deals with accuracy. If you want to differentiate a CH2 and nitrogen, you need a TOF. If you want to differentiate a fragment from your main compound to your successor, TOF is not that much required. You can do it either with a quad or a trap. In both cases, it is possible. So this is the comparison, and this is the compilation table. You can see there are highlighted informations where different differentiation and fragments were very keen, and common fragments were also there. So you need a lot of patience. At the same time, you need a lot of informations about the compound to differentiate that, especially a free radical chemistry or the different rules which governs the fragmentation pathway is important to eliminate the other possibilities to come or to certain the possible predictions. One such prediction is here. The aromatic ring, the common one, the side chain which is not common, the fluorinated thing is also common. So the methyl group can present in the side chain. It is next to the sulfoxides in between. It can also be in the other part of the ring. That cannot be differentiated by MS. That needs again an isolation and confirmation by hetero NMR technique. So impurity profiling is not a small thing to conclude with one instrument or one sophisticated instrument. And of course, this is the comparison and the comparison fragmentation pathway. where the charge retention and the free radical ionization is very much possible in this kind of compounds. The resonance and the inductive effects plays a vital role in proposing the fragmentation pathways. It's very important. And the structure of impurity is finally authenticated like this, and it was since it is isolated, we again spiked and confirmed. At the same time, heteroenamel was also confirming. So once again, the impurity profiling also has the online sophistication now. It is nothing but the mass-directed auto purification, which is also there, which is also now plays a vital role in isolating these impurities, where a mass-based purification is also possible. I will not take much time for this. This is the normal instrumentation technique. You can see here, and it's a sophisticated one where a preparative instrument was coupled with the mass spec. This is again an innovation because it's a need of the hour, because now people wanted to submit the data faster. The competition in R&D is too much that if you are working, even if the patent is expiring next year, people will be working today to do their next level of generic, to get the next level of generic market on its expiry, on the patent expiry, for which they have to submit all the data in time, and they have to solve all the queries in time before it get expired, before the patent get expired. So people want more sophistication, and one novel approach is combining the mass with the preparative system. Preparative, we know very well the traditional isolation technique, but if it combines with an state-of-art mass spectrometry, then it goes as such. In gas chromatography, this is not possible because in GCMS, it's a destructive technique. That's an advantage in the case of LCMS. This is a connections. Okay, it's a normal connections. This is one such example that there will be a delay time. So whoever operates this kind of hybrid technology, it not only applies for these things, but it also applies for the other hybrid technologies. Whenever you apply a hybrid technology, you need to be more careful on the isolation part. This one such automated purification, where you can see here, repurification has resolved these two peaks without in a hassle-free manner. This is another example of this sophistication and the automation in the instrumentations what we have so far. So I think I have covered a bit. I hope I have not bored you, and I have given enough information about the sophisticated instruments on the impurity profiling, because that is more important rather than anything else. Because once you find that in what way you proceed, you will reach the target at an earlier phase. That is what important. That's what today it's technological growth is providing to you. Of course, in few cases it is confusing. I too agree with that. Still, this is needed so that much queries can be addressed in a scientific manner, in a data authenticating manner. That is, which is very, very important in the case of impurity profiling. So the advantage and disadvantage anyway. So thank you, and keep reading all the books, and thanks for the opportunity to meet the fellow scientists of tomorrow, the students and the respective faculty members of JSS College through this webinar platform. And I'm happy to take any questions. Back to you, Krishnaveri madam.
Sir, we got some few questions. I request now Dr. S.N. Mainathan sir to moderate this question session. Is it audible, sir?
Yes.
Yes. கொஞ்சம் close பண்ணிருங்க.
Sir, one first question from the audience. In the case of herbal formulations, which method is suitable for identification of impurities?
Herbal formulations, okay. Herbal formulation means first herbal itself will come as a profile. In that case, herbal formulation needs to be differentiated from ingredients and impurities. First, consistency in profiling needs to be established, no matter it can be an LC based or it can be an GC based, whatever it is. Consistency with respect to the ingredient profiling should be maintained. Then you have to check for the impurities. So in whichever case, it can be a GC base or it can be an LC base. Mostly if it is a intake kind of thing, then it is an LC based. So you have to come under the LC based techniques. Otherwise, if it is an external one, then it is an oil based. In the case of GC based technology will be suitable.
Okay, sir. Thank you, sir. Second question, if we use distilled water, can we avoid the elemental based impurities?
Of course, if you use distilled water, you can avoid, but distilled water, again, distilled water definition itself is changing. Demineralized water or is it a distilled water? Then we have to see how this distilled water is coming. If it is a perfect double distilled or triple distilled water, then definitely this trace level inorganics will never be there. It's not at all possible. But after which, if you are going for a solvent wash, then there is a possibility.
Thank you, sir. Third question, to analyze the low quantity impurity, except LC-MS-MS, which detector is too fit?
See, it is the impurity level or the concentration, what you are looking for decides which sensitive instrument is required for you. Say, for example, if I am looking for a ppm level concentration impurity and it is a water based, then it is LC UV itself is enough. If it is an ointment kind of thing where non-polar liquid chromatographic conditions are coming, then I will prefer to go for GCMS. GC based technique is enough. If you are looking for a concentration in ppb or nanogram scale analysis, then only MS will come into picture. Otherwise, it is LC based UV or a GC based normal analysis in the ppm scale.
Thank you, sir. Fourth one, if tetracycline is degraded, what will be the next form? The broken one will be toxic. Can it be confirmed through LCMS?
Of course, yes, the broken pieces will lead to toxicity only. You know about the reactivity of the nascent nitrogen when in the environment of solvents or in the environment of the clumsy API as valsartan or the sartan families. That's where this nitrosamines are came into picture. That's why this entire sartan family drug manufacturers are threatened by this, especially nitrosamine analysis. And recently, even if you perform the same nitrosamine analysis with respect to GCMS and the through headspace, there is a false positive experiment. False positive was given because of the self-degradation into nitrosamines.
Thank you, sir. Another question, like drug-drug interaction or drug-food interaction or drug enzyme or drug impurity interaction, can it cause any health problems?
Drug impurity interactions will cause health problems. Drug enzyme interactions will be an common interaction which cannot be categorized. Impurity enzyme interaction definitely will cause problem in the DNA level. That's why the impurities will be on categorization of the impurities, the genotoxicity and the carcinogenicity and mutagenicity will be addressed. For that reason only the stringency was given as 0.05 as a nominal threshold and 0.1 as a combined threshold.
Thank you, sir.
The interaction will be impurity with enzymatic based or impurity with the bioprocess based.
Okay. Thank you, sir. Next question, is there any drug impurity interaction in R&D process?
R&D process, I don't think so, because they will, after finalizing then only the process only, they will look into the impurities. Up to that, their target is mainly to take the pure form of their API to 99%. That's what almost all synthetic people will be concerned. The impurity profiling will come into picture at the end of their R&D or at the beginning of the pilot scale.
Next question, will the combination of two molecules play any problem in the human body? Are there more impurities in generic medicines than the branded one? Is it a myth?
It's a myth.
Next one. Sir, what is reporting threshold?
Reporting threshold for what? Reporting threshold means, see, If you say that impurity threshold, that's what the different drug categories, different dosage categories are there. Based on the different dosage categories, this value will change. But for any manufacturers, as far as the online analysis are concerned, anything is greater than 0.05% relative is alarming, and they will have an internal processing, and anything greater than 0.1 is definitely they have to identify and characterize.
Sir, what is enantiomeric impurity asked by one participant?
Hello, I couldn't get your voice.
What is enantiomeric impurity?
Okay, enantiomeric impurity are nothing but chiral impurity. So chiral, there is a chiral analog for a compound or a drug if it has a chiral center. Similar to the other impurities which are process based, there are few impurities which are structurally based, which you cannot avoid. There is a possible and probable formation, and the formation level is very, very high. In that case, the manufacturer will perform a chiral resolution in their process. Though they perform a chiral resolution, there is a possibility for a traces of this another analog of the same product may appear in the drug. That was categorized as enantiomeric impurity. So that has its own values. And in some cases, for example, in the case of losartan, it is 50-50 enantiomer. In that case, the enantiomer is not having any problem. In some cases, where the positive and the negative, that is the enantiomeric positive and enantiomeric negative, the optical isomers, then the combined activity will nullify the response or the drug release or the intended drug applications, which needs to be avoided. For that reason only, there is a resolution process which involved in the manufacturing of the drug itself, based on which few allowable limits will be given for that enantiomer based on the toxicological study or the drug release study. It will be fixed.
Sir, in continuation, we are using chiral column to separate the enantiomers. Whether the impurities also will be detectable using the same chiral column?
Yes, of course. If the impurities are also chiral in nature, then they can differentiate. Otherwise, you have to come through an achiral system for the impurity analysis.
Okay. Does impurity need to be reported in threshold?
Yes, of course. This impurity also should need to be reported in threshold.
Then last but one, that is, how can we differentiate impurities between synthetic and natural products in case of the same product?
That's why the profiling plays a vital role. If it is a natural product, you have to differentiate first the ingredient scale formation, which differentiate the new entity creation or the natural product formation. But in this case, major changes will be polymorphism, because now almost very good synthetic chemists are there to match the entire profiling and blending them as equal to that of a natural combination, the natural profiling combination. There are people to do that, but the major thing is morphology will change. Morphology will play a vital role in such kind of analysis. In that case, impurity will be morphological based, not on the other sexual based.
To limited time, so this is the last question for you to answer. What is polymorphic impurity?
Polymorphic impurity means, see, by structure they remain alike only. By when they become solidify, their angles will be changing. There is a the physical forms will, by if you see it will, you will not see that much difference in that. Slightly crystalline, slightly modification in the angles at which the atoms were distanced. So that comes into picture in the polymorphism.
Thank you.
Thank you, sir, for your wonderful session. Now I request Dr. J.S.K. Nagarajan, sir, to proceed with the next speaker's profile introduction.
Thank you, JP, for the opportunity given to me to introduce second speaker. It's my proud and privilege to introduce the second speaker. Munadi seat. Yeah. He's having 20 years of research and commercial manufacturing as well as management experience in various roles within the pharmaceutical industry. He was working as a head in different company like in India, Arabindo, and in US, New Jersey, Concord Laboratories as well. He was a director for AR and QZ Pharmaceuticals Inc., Stockton, California from 2008, and presently is CEO of Citrogen Pharmaceutical Inc., California since from 2014. He has gained experience in administration as well as management of quality system as well as manufacturing system. And in research, he had a fast-track product development history, and he has filed about 90 ANDAs in 4 years, as well as is having experience and knowledge in regulatory requirements of US FDA as well as other international regulatory agencies regarding product filing. And is more instrumental for more than 100 products filing approvals and USFDA with includes OTC, ANDA, IND, and NDA. And is having knowledge and hands-on experience in analytical technique and instrumentation involved in pharmaceutical companies and research, regulatory aspects, QC, and manufacturing process. And he has played a key role in business transformations of a OTC drug company to ANDA company. He is none other than our own alumni, Dr. Ravi. So I'm just presenting Ravi to you. Thank you. Mr. Ravi?
Thank you, sir.
Yeah. Ravi.
My screen and my voice is audible to everyone.
Yeah. Yeah, Ravi.
Sir, you are audible and you can present, sir.
Video.
And please switch on your video, sir.
Sure.
Yes.
Thank you.
Start.
Sir, you can proceed, sir.
Proceed.
Thank you. Thank you all. I'm very happy and I can't say my words to see you all after a long time. And I'm very proud that I'm back to the institution. Thank you. Thank you for that opportunity, sir. So on today, we are, Dr. Saravanan talked about a lot in the organic impurities and the physical form of impurities. So let's now see about the elemental impurities. So I'm going to talk about the elemental impurities. So segregated the talk in 3 different aspects. So basically, we're going to look around the background about the elemental impurities. And then what are the current global regulatory requirements and a brief overview on analytical techniques. So when we talk about elemental impurities, the first question is what are elemental impurities? Because we have multiple elements in the elementary table. So when we talk about elemental impurities, they are traces of inorganic metals that can be found in a active pharmaceutical ingredient, API, or excipient, or a finished product. So these are commonly known as inorganic impurities. So why do we need to monitor or control these impurities? Because like other organic impurities which can degrade from our drug and it can be active metabolite or other opportunities are there. But these elemental impurities, for example, arsenic or cadmium or lead, these all pose high toxicological risk to the patient. And they don't provide any therapeutic benefit. The biggest problem is in a chronic consumption of this may result in multiple unwanted health risks where we are putting the patient already who is in health-related issues, we are putting them in the health-related risk. And they are very toxic even in trace levels. So when we talk about the impurities, there are certain elements are not considered as elemental impurities. For example, the noble gases, also the reactive nonmetals, because they are already part of our drugs like carbon, bromine, chlorine, or hydrogen, those ones. And the essential metals, sodium, potassium, calcium, magnesium, like those items. So now, the remaining all, everything, like the overall this table, everything is considered as a elemental impurity. So do we need to monitor all these metals for every product? The short answer is no. The long answer is this seminar. So where from these inorganic metal impurities can come into a pharmaceutical? So they are basically multiple sources are possible. So one is the processing tools or aids which we use during our synthesis or manufacturing of drug product. We use compressed air, we use water, we use filtration or purifying aids, drying agents, etc. So these all can contribute or can be a source of elemental impurity to a drug product. Similarly, the processing equipments. So when the processing equipments, for example, it can leach or react with drug or any excipient when it is a direct contact parts, like when we mix the solution or mix the powder. So those type of times we can get leached from the process equipments. And the containers and closures, can again leach metals from its primary contact points. Since, for example, we put injections or parenterals in a vial with the rubber septum. So the rubber septum can leach inorganic metals from the rubber, which is already having it for the vulcanization and other points we use. So those can be a source for the elemental impurities. And we have ultimately the API, our drug substance and excipients in our drug products. So these can also contribute because we use catalysts during the manufacturing process, or we use a lot of mined excipients, for example, calcium carbonate, which is mined from the natural sources. That can be a source for arsenic or lead or cadmium and those ones. So these are the common sources or common known points where we can check the elemental impurities coming to a drug product. So what are the elements we have to monitor, or which elements we monitor? We saw a big list of elements in the periodic table. So this is generally approached by a risk-based approach. So they assess the process and product. So we have to evaluate the possibilities of what element can be present in our product. And we have to verify the market-specific guidelines or pharmacopoeial requirements, because USP requirement and Europe requirement are a little different. So we have to see for which market the product is going. So then we have to establish the controls to eliminate or reduce the levels in the drug product. Also monitor that to assure that quality of the product is maintained throughout the shelf life. So these impurities, how they are being monitored, or what are the common methods commonly employed. So traditionally, we use the limit test for the monitoring of this. Many popular tests we pretty much every day we are using the industry. So one of them is limit test for heavy metals, sulfated ash, limit test for arsenic, residue and ignition, and all these. So what are the problems with this? All these procedures are pretty much very old, like close to 100 years old procedures. And they are known for multiple quality issues. So most of them, most of the limit tests are known for their accuracy issues. For example, these limit tests, we mostly do by visual comparison of color or turbidity. So these color or turbidity comparison is mostly biased and subjective to individual. What looks dark to me is not dark to you or somebody else. So this is becomes a very biased result and subjective. Most importantly, these tests are mostly accumulated limit, like they don't have individual element limits. For example, if we take the limit test for heavy metals, mostly it is 10 ppm or 20 ppm and compared against lead standard. So this theoretically allows higher amount of individual heavy metal in a drug product or API or excipient. So this problem is known for a long time. So the very first public issue on this happened in 1995 when USP published a similar article in Pharmaceutical Forum. So they were describing several problems with this limit test for heavy metals because these are based on the technique of precipitating the heavy metals using sulfide as a sulfide salt and comparing the turbidity. So this one, when they did comparison study among multiple labs in the overall in the global scale, they found that the method was very poor or variable recoveries between the labs and between the analysts, and they lacked the selectivity or the specificity. Also, the volatile elements like mercury and cadmium are lost during the process of testing. So this questionable validity and these issues opened global level discussion or effort to modernize the testing with toxicity-based individual element limits, and also to develop sensitive analytical methods. So following this USP's findings in 1998, European Medicines Agency started to develop a guideline specifically for the common residual catalyst used in the pharmaceuticals. Their main goal was to establish the limits based on the toxicological safety assessments of a common catalytic elements. For example, we use platinum, we use palladium, we use rhodium as catalyst during the synthesis. So they mainly focused on first to develop the toxicity limits for these elements, which are commonly used. So when European medicine agencies EMA were doing their work, parallelly the USP started working with industry and academic people and conducted a lot of workshops and presentations. And so the main purpose of this is revising the heavy metal limit test, which is commonly called as USP General Chapter 231. So this one was led by many academic institutions from worldwide, and as well as multiple industry groups have evaluated. So this resulted in a lot of papers during 2000 to 2008 regarding the limit test for the pharmaceuticals or the elemental impurities in the pharmaceuticals. So in 2008, European Medicine Agency completed their toxicity studies and everything, and then they issued a guideline on specification limits for the residual metal catalyst or the metal reagents, which were commonly used in the pharmaceutical industry. So this was in 2008 itself, they implemented for all the new drugs, pretty much got approved after 2008. All the products went through this guideline. So this guideline, mainly what they did is they introduced a mass-based limit called permitted daily exposure levels. This permitted daily exposure levels are based on the toxicity data, toxicological data they generated on individual metals. So based on this PD data, EMA established a dose-based limit for drug products rather than for the individual drug substances or for excipients, because they considered individual drug products dose plays a major role rather than the individual excipient or individual drug substance, since it can go into multiple products. So they focused mainly on the dose and the route of administration. So in later, what happened in 2008, after the EMA guideline came, USP proposed to replace the General Chapter 231 with two new chapters. So that is called USP 232 and USP 233, which would establish the safety-based limits for the elemental impurities based on the toxicity data from the EMA and the other data, as well as it would also establish appropriate criteria and the methods for the analysis of elemental impurities. With this, all the USP and EMA efforts in 2009, ICH Committee, International Council for Harmonization, they established a working group for elemental impurities in pharmaceutical products. So this group's main intention is to harmonize the technical requirements of these elemental impurities in pharmaceutical products across the major regions. So basically, ICH committee focuses on Europe, Japan, and the US. Even though the guideline is global, their main focus is on these three regions, because these three regions, regulatory authorities work with them. So that is their goal. So in line with the EMA guidelines and USP chapters 232 and 233, the ICH working committee, which is called as Q3D expert working group, they endeavor to set a maximum PDE for elemental impurities in pharmaceutical products. So they mainly focused on oral, parenteral, and inhalation routes of administration based on the existing toxicological data. So what happened in June 2013, ICH came up with the first draft version, which is called ICH Q3D guideline, which talks about the elemental impurities. That was opened for the public review and comments. So during 2014 to 2018, when the guideline was in the comment process, major international pharmacopoeias like USP, European Pharmacopoeia, Japanese Pharmacopoeia, British Pharmacopoeia, and other local industry representative bodies, Pharmaceutical Manufacturers Association, Consumer Pharmaceutical Association, and other major international regulatory agencies, FDA, EMA, and everybody discussed with ICH and harmonized the pharmacopoeial and regulatory guidelines along with the ICH guidelines. So in January 2018, USP officially enacted the revised chapter of 232 and 233 and depreciated the old limit test to 231 effective from January 2019. So right now, the limit tests are not official as per the USP, so it has to go through the USP 232 and 233 chapters. So the latest ICH Q3D guideline is a revision 1 version, R1 version, so which is issued on March 2019. So now we all know that it is impossible to assess or monitor a drug product for every element in the table of elements. So the elements to be monitored are selected based on their toxicity, the permitted daily exposure limits, and their likelihood occurrence in the drug product. So this was done by ICH, and ICH derived these PDE limits based on the no observed effect level or the lowest observed effect level in the most relevant animal study models. So based on that, they have, with the PDEs, they have identified a total of 24 elemental impurities as critical elemental impurities that are to be monitored in a drug product or in a drug product components, and they have specified their maximum daily limits or maximum permitted daily exposure levels, so in a microgram per day. So for all the delivery, pretty much the oral, inhalation, parenteral, the major drug delivery categories, they have assigned the limits. And further, what they did is this 24 impurities also they classified into 3 major categories based on their toxicity and their possibility or likelihood probability to present in the pharmaceuticals. So that way we don't need to monitor all 24 impurities at the same time. So they have classified and given a requirement-based or risk-based approach to monitor these impurities. So the classes which they put together is they put together in a 3 classes. So class 1 is they identified 4 impurities. Basically, these are heavy metals: arsenic, cadmium, mercury, and lead. So these 4 elements are very well-known human toxicants, and they have limited or no use in the manufacture of pharmaceuticals. But their presence in the drug product typically comes from the commonly used materials. For example, from the mined source excipients or derived excipients like calcium salts or calcium or other sodium salts, which are mostly from the mined sources. And then it can be leached from the packaging components, since we use a lot of metals to stabilize the rubber or neoprene or other closures. So those also can leach these metals into a pharmaceutical product. So the class 2 are the elemental impurities, which are generally considered as root-dependent human toxicants. They are further divided into subclasses as 2A and 2B based on their relative likelihood occurrence into a drug product. So in class 2A, they put 3 elements: cobalt, nickel, and vanadium. So these elements are having high probability of occurrence in a drug product, because cobalt or nickel or vanadium can come from process equipment surfaces. Like, say, for example, stainless steel can leach the nickel. So that's how they put together these 3 elements in the class 2. So these 3 elements have, along with the class 1 elements, has to be assessed across all the products or all the routes. So this 7 impurity is collectively called as Big 7 in the industry. So this 7 metal products we have to risk assess and we have to monitor, so that we have to do it. So the class 2B is 10 elements. They have reduced probability of occurrence because they mostly come from only when you use it as a catalyst or when you use other materials in your formulation that has been used this as a catalyst. So therefore, they have excluded this from the risk assessment, which has to be not compulsorily has to be done, only it has to be performed when we intentionally added during API synthesis or drug product manufacturing or excipient manufacturing. The class 3, these are 7 elements. They have relatively low toxicity by oral route. That is, they are more than 500 microgram per day limits. So they are not considered as a risk for oral route, but they may require to be considered in the other routes like inhalation and parenteral routes. So for oral routes of administration, we don't need to monitor or risk assess these impurities unless we know that it has been added to API or excipient or in a drug product. But for parenteral and inhalation, no matter what, we have to do this elemental impurities to be risk assessed. So they are, if their limit is less than 500 for that particular routes. So apart from these 24 elements, any element, if the manufacturer or API of or a drug product or an excipient, if it has been used in the process, then we must assess or check those specific metals. Say, for example, iron, if we use a color based on iron, then we have to see that the iron adsorbed colors are within that limit. Same applies to the aluminum lake colors. So these are becomes the basic guideline to what we have to look for or how we have to risk assess. So the limits are I just gave it in the table. So these 7 impurities are cadmium, lead, arsenic, mercury, cobalt, vanadium, and nickel. So these 7 metals, whether we add it intentionally or if we don't add it intentionally, we have to do it risk assessment. for all the drug products. So the class 2A, if we don't add it, we don't need to do any risk assessment. Only if when we add it in an APA or excipient, only that time we have to do the risk assessment for these class 2A and 2B and class 3 elements. Some of the elements like which are very toxic in certain routes, say for example, the lithium, antimony and the copper, they are less than 500 microgram per day in parenteral route. So these has to be assessed when we develop a parenteral route along with these 7 elements. So similarly for inhalation drug product, we have to monitor all these impurities, the antimony, barium, molybdenum, copper, and tin, chromium. So these ones when we do for the inhalation products. So how do we assess the risk of these metals in a pharmaceutical product? So how we do that? So the risk assessment, it is a systemic process to evaluate the possibility of the risk that these elements, whether it can be introduced in a drug product or can be exceeding the PDE levels, which we have seen in the previous table. So typically these are established during the R&D stages before the commercial manufacturing. So the risk assessment process involves 3 major steps. So the first step is we have to identify the known and potential sources of elemental impurities that may find a way into the drug product. So what are the components of the drug product? So excipients, APA, drug, and drug substances. So that can be bringing the elemental impurity to the drug product. Similarly, the facility, manufacturing, packaging, these can bring the elemental impurities to the drug product. So during this risk assessment, the potential contributions from each of these material and each of the process steps, we should evaluate to determine the overall contribution. So typically in the industry, what happens is we split this in 2 portions. So the facility, manufacturing, and packaging, these are common items we share across multiple products in a facility. Say, for example, we have a liquid oral facility. So the reactor and packaging components which we use for multiple products, and the facility where the water and everything which are common in a facility. So these can be a common risk assessment during the overall facility establishment or a part of GMP process. We can periodically look into it, and then we can create a separate risk assessment possibility from these ones. Whereas the excipients and the APA, they are the most common sources which majority of products get the elemental impurities. They can be controlled by GMP process like vendor qualification and selection of process to keep the tighter controls. So in general, so the product or formulation specific risk assessment is done for each product, and then common risk assessment specific to the manufacturing unit shall be established and reused and re-verified over a period of time. So in the step 2, what we do is we evaluate the presence of particular elemental impurity in a drug product, either by determining the observed or predicted levels. For example, I use 10 components, so we see what are the reported levels in the 10 components by the manufacturers, and then using that to do a risk assessment. Okay, so this has only 2 microgram, this has only 1 microgram. Can this, if this I added it in this percentage level, can it go beyond my allowed PDE limit, or can it increase? So those changes, those evaluations has to be done. So in the step 3, what we do is we identify if controls are built into the process. For example, we find that we are getting an elemental impurity from the water. So then we should put a control to that to eliminate or to reduce that level into the drug product. So, and then all these are summarized as a part of product development report in the risk assessment section. So usually this becomes a part of drug product approval when we file it to the regulatory agencies like US FDA or MHRA or European agencies. So this becomes part of the filing documents. So for example, here let us assume a theoretical case. We have a drug product which is made with some drug substance we put, and then we add excipients, microcrystalline cellulose, lactose, calcium phosphate, magnesium stearate, everything we add. So now let us see what are the risks or what are the potential elemental impurities based on the risk assessment. So the dose I have given here for information. So the intentionally added catalyst or others during the synthesis or the purification of the material. So drug product reports say, for example, palladium and nickel. So none of the excipients say, okay, we didn't use any catalyst or other metals. So now the palladium and nickel becomes your risk assessment. And then potential elemental impurities with a relatively high abundance are which one we have to compulsorily monitor. So that becomes class 1 and class 2A anyway for the drug product. So individually, since the class 1 is most important or most highly toxicant, we have to monitor these in the individual drug products, individual components of the materials. So then potential elements or impurities from the manufacturing equipment. So that we have to evaluate based on the process. Say, for example, okay, whether this material can complex or leach from the manufacturing equipment when they are in contact. So based on that, we have to do it. Since we use mostly the stainless steel equipments in the industry, most of the times nickel and vanadium are commonly comes from the potential elemental impurity from the manufacturing equipment. So now the next one is potential elemental impurities from the container closure system. So this is, we said, as a oral tablet product. So when a oral tablet product, it either goes in a HDPE bottle or alu pack or blister or some kind of those things. So in a solid state, there interaction between a packaging material to get the elemental impurity from the packaging material is very minimum or almost not there or not reported. So that becomes none. So here if we see, we have titanium dioxide and iron oxide. So the titanium dioxide and iron oxide becomes again metal which we are introducing. But these ones are not added into the risk assessment because the titanium oxide and iron oxide levels are less than the required PDE levels. So that means these can be eliminated or it can be not required to do the risk assessment. So in summary, if we see the risk assessed elements which we need to monitor are 4 class 1 elements: arsenic, cadmium, mercury, lead, and cobalt. Then 3 of the class 2A, cobalt, nickel, and vanadium. And then we have used the catalyst of palladium in drug substance. So that one also has to be risk assessed. So that's how the risk assessment is typically built in industrial environment or during the development or during the initial stages of drug product scale up or manufacturing time. So now we have to design or develop the controls. So how we do that? So typically these controls are established using by the quality by design approach. So in this case, what we do is all components, API and excipients, are analyzed at batch levels, and then they are controlled based on the risk assessment process as individual manufacturers of the API and the excipients. So these limits are at which we control them is tighter than the drug product limits. So these limits are recommended by guidelines, so that I didn't give it here, the table. So if anybody is interested, they can refer the USP General Chapter or the ICH guidelines. So the varying quantities in different drug products can be handled by this tightened limits. So say, for example, I need to use MCC more in a product. So when I control the MCC's elemental impurity in a lower level, so I can use higher amount of MCC in a drug product. So that's the basic logic behind the tightened limit for the individual components than a drug product. Then all processing equipments are designed or modified based on worst case scenario examples or approaches. For example, yeah, when we mix an acidic solution, say for like citric acid or anything in a stainless steel, they can leach nickel or cadmium like vanadium like impurities from the stainless steel surfaces. So we can employ glass-lined reactors to avoid these. So, and also we can include incorporating in-process monitoring steps when a risk is identified. So that's how the QBD approach we do for the processing equipments. And then the containers closures, we have to evaluate the container closures for the leachable studies. That is apart from the elemental impurities, there is other impurities also we have to monitor. So the leachable studies are very important with the containers and closures with respect to any liquid products, specifically oral or injectables. And if any risk we found, so we can use we can employ alternative packaging components. The most common ones are like form fill seal techniques to avoid the rubber septums. So those opportunities are available. So use of this alternative process or designing alternative dosage forms whenever it is possible. So that minimizes the risk. For example, the form fill seal bottles. And also the other approach which industry does is so sometimes we switch the formulations instead of liquid injectable, we move to the powder for injection so that we can improve the product stability as well as avoid these leachable impurities from the stopper. So that is also another opportunity which we commonly employ during the design controls. So now we have to put the control. So now we need an analytical method to do that. So analytical method, we have to do it. And then the important thing is how we do it. So there are three options. So one is drug product analysis option, and another one is summation option, and then individual component option. So in the drug product analysis option, it is typically what we do is we individually analyze the dosage form and then scale the limits to the maximum daily dosage unit, or we have to convert that limits in comparison with the microgram per day limits to the which is prescribed in the guideline, and then compare that whether it is less than or equal to the daily dose PD. For example, the drug product which we talk about, so we measured those drug products. So these are the values we got in microgram per gram for each of these elements. So the maximum daily dose, what we see is it was 2.5 grams per day. So we have to multiply this with the 2.5 gram. So that becomes microgram per day. So the calculated microgram per day values are less than the limit given by the USP or the ICH limits. So then the result becomes pass. So that's how the every drug product it is done. So the key here is the maximum daily dose. So depending on your daily dose, your product limits will vary. Say for example, the same drug product, I have two different limits, two different dosage levels. Then the worst case I have to take. So the maximum daily dose should be considered on the worst case scenario to maximize the controls. So that's how it is usually done. So the second option is summation option. So in this option, we say that, okay, we have established all the controls and we now control our impurities at the drug product or drug substance or the excipient manufacturers. And we get that material, we individually test them, and our process doesn't contribute any of the elemental impurities. So that once we develop that, then we can just go with the summation option. We don't need to do any analysis. We just get the individual certificates from the API and the excipients, and then each of the components of the drug product, we just take them and then we sum all those impurities and compare that against the daily dose PDEs. So since this is only a paper-based approach, a complete documented evidence should be there to prove that elemental impurities cannot be contributed by the manufacturing process or container closure system. So usually this is done during the production validation stages. So the product validation, when they manufacture, they establish all these documents saying that different stages of processing we analyzed, we found no elemental impurities have been contributed by the process. And then with that documentation over the period, the commercial manufacturing quality control uses only the summation option. So there is another option which is prescribed by USP basically. So when your daily dose is less than 10 grams, if your all drug substances and excipients meet the stringent limits given for the lower limits, which we talked about earlier. So those limits, if it is within that, and then these components can be used in any level, if it is oral product and if it is a less than 10 gram dose. And similarly, we can do that for the parenteral as well as the injectable products, but their individual limits are different from the oral limits. They are titled limits. So that's how the individual component option works. So now we know how to control and how to calculate. So the important thing is now we need what method we can use. So what are the recommended methods in the guideline or in the pharmacopoeia? So that is, if you see ICH guideline, so generally they don't recommend any specific analytical technique or procedure. So individual pharmacopoeias, they recommend the analytical methods. Mostly all the USP, European pharmacopoeia, Japanese pharmacopoeia, all three of them recommend the ICP-based technique. So ICP OES and ICP-MS. So the inductively coupled plasma optical emission or mass spectrophotometer. So these ones are commonly recommended by the pharmacopoeia. But so when you use these pharmacopoeial methods, you can do a minimum validation because they have given you the method conditions and everything which has been established. So those ones are typically the minimum validation which you do for these pharmacopoeial methods is basically the specificity, accuracy, and robustness for your drug product. So alternative analytical methods, yes, not every company has the ICP OES or ICP MS. So can we use any alternative methods? Yes, pharmacopoeias allow the use of alternative methods apart from the ICP-based techniques. But only this is, you have to completely validate. And what are the validation parameters we have to meet also being prescribed in detail by USP. So they give three different classes. So if when you develop an alternative method, which is say for example, you improve the limit test method itself in a better way, you eliminated or your need is fulfilled by the limit test, then you have to validate that for the specificity and the limit of detection. Minimum of 80% limit should be detected by the limit test. And it should be specific to the individual metal. So we have to prove that with, in presence and in absence of metals, how it works, and this one. So same way you can develop a limit test based on the instrumental approaches, for example, fluorescence detection or UV-based derivatization methods. So those one also can be done. So in that cases, you need to do the specificity, precision, and the limit of detection. Again, here also in the limit of detection, your method should be detectable for at least 80% of the limit. So that means if my limit for lead is 5 microgram per day, my method should be able to detect at least 4 microgram per day. So if it is not that, then the method cannot be validated, or that alternative method has to be improved to meet this requirement. So when we do a quantitative alternative method, say for example, we did a HPLC method for lead analysis. So that can be also validated and it can be used. So the accuracy should be 70 to 150%. These limits are a little bit different from the general ICH validation, this one's, because they consider this as a special case. And this one limits are a little bit different, and it is controlled at the chapter level of this impurity chapter 233. So 70 to 150%, and the recovery has to be proved at 3 different levels, minimum 50% to 150%. So each are done at triplicate samples. So basically you do 9 samples over the range. And the precision, yes, obviously the repeatability should be not more than 20%, and the intermediate precision between the day or between the lab or between the analyst or between the instrument, it should be not more than 25%. And then obviously we have to prove the specificity and the linearity and range. It has to be a minimum of 50 to 150% of the target. And then limit of quantification, when it comes to the quantitative methods, it should be minimum 50% of the limit. So that means if we have 5 microgram per day limit, it should be minimum 2.5 microgram per day, should be quantifiable with the alternative method. So that's about the method. So now about the sample preparation. The sample preparation also USP recommends the procedure. The reason why the sample preparation is very important in the elemental impurities is because as long as the sample directly dissolves or directly already as a liquid, you don't have any problem. But when the sample is not completely soluble, you need to prepare an indirect solution. For example, microcrystalline cellulose, it will not be soluble in an organic or any aqueous solution. So that time we need to perform a total metal extraction. So the biggest problem with this total metal extraction is so either it can precipitate for depending on the salt, because we need to add some acid or anything and to get that. So different salts, different procedures can lead into the loss of elements. For example, mercury can be a little bit volatile. When you add some carbonate salt to it, it just gets evaporated. So that's the critical process here. So for this indirect solution preparation, USP recommends a closed vessel digestion procedure. So the closed vessel digestion procedure is nothing but a microwave digestion process, similar to our household microwave. The microwave heats the sample in a closed container. Then from that, the liquid is being taken and used for the analysis. So that way the volatile components don't escape from from the sample preparation. So that's the common sample preparation. If the sample is directly soluble in aqueous or organic solution, we can directly use it without any sample preparation. So as long as it can be analyzed without any interference. For example, when we use aqueous solvents, many times the buffers can precipitate or interfere with the analyte of interest. So that has to be taken care when we prepare the samples. So what are the common analytical techniques we can use for the elemental impurities? There are multiple techniques. So popular technique is ICP-based techniques. So the ICP-OES and ICP-MS are the commonly used technique in the industry. So the LA-ICP-MS is a recent development. So basically what they do is they use the laser ablation to get the elemental vapor from the samples. So this is a pretty new technique. The most important problem in this technique is we did evaluate this instrument for quite some time. But the biggest problem is there are not enough standards have been developed properly for this one. The commercial standards are not much working into this. So probably in 2 or 3 years, this may become a mainline instrument in this one in the analysis field. And the other common technique is X-ray fluorescence. So there are 2 types of X-ray fluorescence, wavelength dispersive and energy dispersive. So that is quite interesting technique. I will just give some introduction on that technique in the later slides. And then there are other techniques, the neutron activation analysis, spark source mass spectrometry. Though these 2 techniques are extensively used in the forensic and environmental analysis, not much is being used in the pharma applications, but still they can be explored to the applications. So that's one. And the other one is microwave plasma atomic emission spectroscopy. So that is also another technique which commonly used in the environmental industry, but not be used much in the pharma. Apart from the ICP in pharma, we use extensively the atomic absorption spectrophotometer. So those ones. But with respect to the elemental impurity analysis, there are certain challenges with atomic absorption. One is the sensitivity. So that is one, this one. But alternatively, that can be done or managed with the standard addition method or something like different approaches like that. The other major challenges is multiple methods. So we have seen 8 metals now. We have to see in example drug products. So in the atomic absorption, then it becomes 8 methods. So that is one challenge. And the sample quantity, since we do 8 different methods, again, the sample quantity, because the limits are at lower level, so you need a higher sample quantity to make the atomic absorption to get sensitively detect the levels. So that's one. There is another very recent technique which is using the metal-specific DNA ligands. It's quite interesting. We did evaluate this technique, and it was really working good in the water, plain neat water elemental analysis. So we are trying to optimize this for the pharmaceutical purposes. So this is like fluorescence spectrophotometer. So just you pass the sample through the DNA specific ligand filter. So the DNA binds to the element, and then it gives the fluorescence specific to the metal. So the challenge is not many kits available commercially, and they're very costly. So possibly in the future, with the applications are expanding, it can be going down with the price, but for exploration purpose, yes, it can be definitely used. And the other thing is the ion chromatography, or the HPLC. So this is pretty commonly available in every industry. So HPLC can be very well repurposed for multiple users. Only our imagination is the limit here. So we can use multiple detection techniques. We can use multiple modes of chromatography. The biggest challenge are this one is the sensitivity, because we have to see how much we are diluting the sample, how much sensitivity we need, because already our limits are on the lower scale. So we have to see whether we have to do any derivatization or anything to improve the sensitivity. So those are the challenges. And the other challenges is the interference or separation from the organic matrix. So like we might be having organic impurities, we might be having the API, or we might be having the excipients and all these stuff. So we have to develop, or we have to optimize the interference and the separation from these organic matrices. The sample quantity and the method optimization is the biggest challenge in the HPLC, but otherwise it can be done. It is not that it cannot be done, it can be done. So the ICP-based technique, it is basically what happens is the sample solution enters into a spray chamber. So that is carried by a gas. Usually the argon is the most common gas it is carried. And then it enters into the ICP touch. ICP touch is nothing but a plasma which is maintained at high temperature, approximately about 10,000 degrees centigrade. So which ionizes this argon and sample mixture. So that goes through the detection technique based on the, if you are OES, then you have the optical emission, or mass spectrophotometry if you are having mass, and then that is detected as a metal when it comes that. So basically the basic principle here is if the element comes in the ground state, when it enters to, when it combines with the energy of plasma, it gets excited. So because of this excitation, yeah, fluorescence or optical changes happening in the elemental level, that is individually characterized for each element, the wavelength or the wave numbers. So depending on that, we separate the elements. So that's how it is being done. So the OES and AES are pretty much same technology. So for here, for the discussion, I'll keep the ICP OES and the AES represent the same, because going into that detail, it needs another whole session for explaining the OES and the AES. But otherwise, it is in common. You can consider as the same principle. So when we look at the ICP OES, this is the typical layout of the operating principle of the equipment. So basically, we have a nebulizer and a spray chamber, which powered by pressurized by argon gas. So your sample is pumped through a pump to this nebulizer, the spray chamber. So with the nebulizer and the argon gas, it creates a vapor, and that vapor enters into the ICP torch. So there, where the elements are get excited with the energy from the plasma, and that is detected by a spectrophotometer. So that with the further conversions in the electronic, this one we see it as a spectrum or a graph in the software. So that each of these elements have characteristic fluorescence spectrum. So that is the main key or the principle which we detect in this. And these responses are specific to their quantity, so that it gives the linear response. The biggest advantages with the ICP method is the simultaneous analysis of multiple elements can be done in a single run. And sensitivity is up to nanogram per ml. So these two are the greatest advantage. But the disadvantages is it needs typically a controlled environment, because the argon gas cannot be inhaled or cannot be exhaled to the environment. So you need a separate controlled environment for the handling, as well as the equipment is so complicated, and the cost of equipment is also high. And also the cost to perform analysis also high, because you have multiple accessories and consumables, which are adds the running cost. Other common advantage with all the elemental based methods are the matrix interference from high concentration elements, like when we have a excipient calcium based, or when we have a excipient as calcium carbonate, or any API, inorganic based API. So those matrix influence are very high, because the elements are emitting a characteristic fluorescence or characteristic wavelength modifications. So when one element is in very high concentration, it creates a matrix impact or a matrix interference, so that the minor elements fluorescence spectrum signals gets weakened or diluted, or happens in a band spread. So this matrix interference is pretty much common in every inorganic analytical method. So that has to be taken care during the development or during the method establishment process. The ICP-MS, pretty much it is same as the ICP-OES. The major differences is the detection part, where it is a typical mass spectrophotometer like quadrupole or time of flight. So mostly they use the quadrupole one, so that's the one which detects. So basically they filter the record mass for each of the element to be monitored, and those are monitored. So similar to ICP-OES, this is also sensitive, and a little bit sensitive, higher sensitive than OES or AES. So they can go up to picogram per ml, and the simultaneous analysis is possible. Similar to OES, this also required controlled environment, and cost, everything is pretty much similar to the ICP-OES technique. So the other technique which we commonly use in the industry is X-ray based fluorescence spectrophotometer. So there are two different techniques are there, wavelength dispersive and energy dispersive. For the discussion, I'll just give the overall brief, only the principle. So what happens is basically an X-ray tube beams X-ray to the sample. So the sample pass through a collimeter, and then it gets into the detector. So that's how the thing is happen. So how it detects this is when the X-ray hits any element. So we all know that we have K shell, L shell, M shell. So each of the X-ray, it hits one of the electron from the element. So because of this, it creates an excitation state for the element, so which makes the element to emit a fluorescence or create a fluorescence spectrum. So with that, we maintain. So usually the alpha lines and beta lines and gamma lines are based on the which shall get excited. Based on that, you get. So basically, this is a typical spectrum, and we run an X-ray fluorescence. So the biggest advantage with this method is the sample preparation is very simple. We don't need to go through all that closed vessel digestion using concentrated acids and all those stuffs. We can just directly keep the tablet or API powder directly, and then we can measure the sample. Just we need a uniform particle size for the just measurement. And pretty much this is also simultaneous analysis, and cost of analysis as well as instrument is very cheap. And the operation is very simple. If you know to operate UV, we can operate this instrument. So that is that much simple instrument. But the disadvantage is also there. It is sensitivity is only at microgram levels. So if your dose is very low, say for example, if you have less than a 1 gram dose, then your limit becomes much smaller. So that becomes a little bit difficult or disadvantage on this instrument. If your dose is like 10 gram, then or if your dose is 1 gram, then you can definitely do it. So the higher the dose, your limit goes down. So when it goes more than 10 gram doses or 20 gram doses, the limits becomes so small numbers of in the microgram or picogram levels. So that time the sensitivity is very difficult to achieve in this technique. Also, this also has the matrix interference, but there are, as I said, it is common with every elemental impurity analysis method. So that is something which we need to do it, but there are a lot of ways to get resolved that. So definitely this is a good technique to evaluate and read and know much about this. So in summary of all the elemental analysis techniques, if we see, because the overall goal is ultimately at the end of the day, you need to use some analytical technique. So which analytical technique to use? The answer is no single technique will give you everything. So every technique has its own advantage and its own disadvantages. But obviously, we have to, based on our need and based on the application, we have to select the technique which we need. So as we have seen, the ICP-based techniques, they are low detection limits, and they have multiple elements can be analyzed at the same time. And even in MS, we can do even isotope level analysis, but the disadvantages are the cost and the matrix effects are pretty much common. So in the OAS, we have to do only a liquid samples and both cases. So that is also another disadvantage. In the X-ray based methods, yes, we can do the solid and liquid samples. We can eliminate the sample preparation steps. So that's a big advantage, but the disadvantage is detection limits are too high. So for certain dosage limits, it may not work out. So we have to see how we use that based on the application. Similarly, the AAS or Newton activated analysis or other techniques, they are low, their detection limits are low detection limits are possible. And, but the problem is they are very time consuming because they all have to go through one by one metal. They are not simultaneous approaches, and they are having their own this ones. So other techniques like HPLC or other ones, we need to develop our own custom development, but it can be done. It is not that it cannot be done. Only it needs the time and resource to do that. Obviously, it is versatile because every lab has the HPLC, so that way it is wide availability and versatile. Disadvantage, we had to see the sensitivity, but there are multiple derivatization techniques using the fluorescence tags or UV-based ligands, so to improve the sensitivity, so that can be done. Interference or separation from the organic matrix is the major challenge, but that can be done with the current advances in the solid phase extraction cartridges and those ones. It is not that unachievable, but only the dedication and the time we need to put together. And every drug product needs this to be repeated. So that is another thing. So that we have to see that. The sample quantity and the method optimization can be a challenge or a disadvantage in those techniques, but it is not a stopping point for sure. So that can be definitely evaluated. So with this, I believe I have given an overall view about techniques. Thanks for all your patience. If there are any questions, I'll be happy to answer.
Thank you, sir. Now, Dr. S.N. Menathan, sir, will moderate the question session. Hand over to Menathan, sir.
Thank you, Dr. Ravi. Able to hear?
Yeah, yes, sir. Yes, sir.
Okay. How are you?
Good, sir.
Nice to see you after a long time.
Yeah, my inorganic teacher is moderating me.
Nice. I won't ask pass or fail.
That's
Okay.
why I said it is subjective.
Okay. Saraj, there are some queries from the participants. Okay. The first question, what are class 2A and 2B impurities?
Okay, class 2 and 2B impurities are commonly comes from the process equipments or the process catalysts. So those are the class 2A and 2B. So they have multiple chances to be present in the drug product or drug substance or excipient. Impurities are there. So in class 2A, they have mainly put together the impurities, the three impurities, cobalt, nickel, and vanadium, which comes from the process equipments commonly. So those are the class 2A impurities. And class 2B impurities are mainly arises from the common catalysts. So there are 10 impurities are there in the class 2.
Next one, what strategy we should adopt for isolating and quantifying arsenic impurity in natural product based?
The natural product, the best thing is for the natural products, because the natural products, the biggest problem is there are two ways here. One is the analysis part, another one is the purification part. The purification part, when we talk, the best technique to purify is for the arsenic is column chromatography based using ion exchange resins is the best technique when it comes to the arsenic purification. When it comes to the quick and dirty technique, it is the partial crystallization using sulfides or others to precipitate the arsenic, but they may not result that much pure. But if we know that, okay, we have 100 ppm of arsenic, we want to bring it down to 50 ppm, yes, that can quickly work. But from 100 ppm, if you want to bring it to 1 ppm, definitely we need column chromatography based or preparative chromatography based purification using ion exchange resins. For analytical purpose, any of these analytical techniques will work, either ICP-MS or X-ray fluorescence or ICP OES. Only thing is from the sample preparation perspective, X-ray will be easy for a herbal product because we have multiple components. We have to optimize the sample preparation for the ICP techniques might take little difficulty.
How to make an elemental impurity analysis for metal-based herbo-mineral formulation in Ayurvedic or Siddha medicines?
In Ayurvedic or Siddha medicines, the common problem is so what are the metals we added, or what are the metals we believe it is for the therapeutic purpose which we intentionally added. First, we need to identify those, and we have to make sure those are not additionally contributed by other components. So that is the first and important thing which we have to look into it. And the second thing is when it comes to the analysis part, we have to see because the Ayurvedic and herbal medicines, they don't have a separate limits or guidelines prescribed. You can in general, you can take the dietary supplement guidelines, which is a different guideline with a little bit different limits based on the consumption. It is not based on the dose. It is based on the consumption. So that is, they classified it as a food. If you are taking for a long time, how much limit you can use. So those ones are pretty much good place to start for those kind of products.
Thank you. Next one, what is the impact of elemental impurities on biotechnology derived products?
It is same as the pharma products. So when it comes to the biotechnology derived products, the major interference or major source of elemental impurities from the digestion or the culture medium where we grow the organisms to produce the proteins. So, and a lot of proteins will have metal specific scavenging or complexing property. So that is the biggest challenge. So usually this is addressed during the production itself. When we develop the process for biotechnology derived products, we take care of those steps during the development itself. So that way it is equally important as a pharmaceutical product. But only the advantage with the biotechnology products are most of the biotechnology products are very low dose. So you have high limit of acceptance for those. For example, 10 microgram or 20 microgram only your daily dose. So that gives you a larger room to relaxed controls.
Possible to eliminate 100% elemental impurity?
It is possible, but whether that is cost effective or not in a common process, that we have to see, because catalysts and these ones are very common in many of the synthesis routes. So that ones are pretty common in the sense we may not get a catalyst free material. We can definitely do it, but it's gonna take the cost.
Okay. Next one, how to frame the linearity for the elemental impurity, especially based on the quadratic plot or weighing assessment?
The most accepted one is linearity based on the relative square deviation. So that is the linear regression is the most accepted ones. But when you have very low concentration and your method has different ones, definitely you can use alternative techniques like scattered plots and other techniques. So it is not hard and fast that you have to use only this technique, but it depends on the case-to-case based scenarios.
The last question. Can the spin orbital coupling of composite material can be identified using X-ray fluorescence spectrometer?
Not. X-ray fluorescence spectrophotometer cannot identify that. It can do the thickness or it can do the total metal, but not at that level. So probably for that, maybe you need like isotope-based analytical methods, or you have to go into the photon-based techniques like XPS and those techniques.
What about any patents are there for this kind of elemental impurities?
Elemental impurities are generally known as there are multiple patents on the instrument detection techniques and instrument principles. So that is a different scenario. So as a impurity alone, it doesn't have any patents, like, because these are common elements which is known for multiple years. So yeah, process-based patents can be developed for individual APIs or individual excipients. Say, for example, you have the classical example I can give is the lanthanum carbonate API. So it is a 100% inorganic API. So there are multiple patents on that with respect to which talks about the elemental impurities, because the lanthanum is mostly from the mined source. So those purification and those ones are there multiple cases. So they are product specific and non-obvious patents. So in general, overall, if you see about the elemental impurities, quantification or control process is pretty much specific to every industry. That is, making a patent on that is, unless you are an instrument manufacturer, if you are doing a quantification technique or a detection technique as a patent, then it makes sense. Otherwise, it doesn't make any sense in the pharma environment.
Thank you, Ravi.
Sir, thank you, sir. I'm Dr. Jayaprakash. I have a question. Generally, in organic impurities, we have a genotoxic, carcinogenic, such a classification. In elemental classifications or any kind of specific
Yeah, that's the class 1, class 2, class 3. So the class 1, class 2, class 3 are defined based on the limits of toxic levels. So basically the class 1 is the highly toxic impurities, like your genotoxic impurities. So those 4 metals, arsenic, cadmium, mercury, and lead are the most toxic metals in the classes. So the class 2 and the class 3 are, class 2 is not commonly present in the pharmaceutical product, but it can come into the product when we use it as a catalyst or from the process equipments. So they are a little bit higher limits, but so when you have to monitor, like same like your degradation product. So if you find a degradation, you have to monitor. If you don't find, if you don't need to monitor. So when you use, you have to monitor. If you don't use, you don't need to monitor those impurities. Only the class 2 is related to process equipments, you have to monitor no matter what, and along with the class 1. Otherwise, class 2B only you will monitor if you are using the catalyst or the metal as an excipient. The class 3 is route specific. So basically in the oral category, the class 3 is much safer because their limits are very high. But in the oral, in the inhalation, it becomes very toxic. So some of the class 3 impurities inhalation route limits are less than the class 1 limits because their toxicity levels are that much higher when it inhaled as a impurity. Same way in the parenteral. For example, lithium. Lithium is even used as a therapy in the antipsychotic. But when it comes to the impurity level in the drug product, on the parenteral products especially, it has to be tightly controlled because it can directly be bioavailable to your blood and can cause a lot of impacts. So basically, this is route-specific limits and toxicity-based limits. Both combined together, it comes as a class 1, class 2, class 3. So that's why you don't see the genotoxic or highly toxic levels. So basically, the class 1 is highly toxic. Class 2A is next to that. Class 2B and 3 are subjective to the route and subjective to the process.
Thank you, sir.
Good evening, sir. This is Dr. Babu.
Yeah, Babu.
Good evening, sir. This is Dr. Babu.
Yeah, Babu.
Yeah, I do have one question. What is the scope of MS alone, LC-MS alone, in these elemental combinations? because we do understand that, one of the problem is the adducts which do form in the MS. So keeping this in mind, what is the scope of the MS in elemental analysis?
See, MS as a detection technique, yes, it can be used for monitoring the elemental impurities. But here, one thing you have to remember, the mass levels are going to be very low level. So in general, the LCMS instrumentation is not designed for this kind of application, but it doesn't mean that we cannot do it. It can be done, but a lot of development effort and optimization effort is required, because you need to separate the metals in the LC part and detect it in the MS part. So when you separate the elements in the LC part, so there are two options to you. Either you can use the ion chromatography based technique like ion chromatography columns, or you can use the other techniques like ion pair techniques. So when you use the ion pair techniques, the problem is going to be on the detection side, because you might be having a lot of salt contents in the ion pair techniques. But you can do it with the tetrabutyl ammonium hydroxide or something like that, liquid-based salts where they don't precipitate in the MS. But the challenge is you have cations, you have anions, both are there. So in a single run, you have to separate them and you have to quantify them using the mass. So it can be done, but it is not can be done easily. So it has to be focused effort has to be initiated to do that. Definitely it can be done. It is probably the best one I will suggest is LC and fluorescence based detector, because the advantages is when you use the LC and fluorescence base, you can go with the post-column derivatization using fluorescence ligands, which are very widely available and which is known to be very sensitive to a lot of elements. For example, lead, cadmium, a lot of environmental applications are there with the post-column derivatization. So those ones are definitely much better compared to the LCMS based. If it is LCMS, yes, definitely you have much advantage, because with the time of flight and ion separation techniques based on the mass can be used. Just with the LCMS, it might be trivial, but it can be done.
Last question.
Yeah, thank you, sir. And the last question from our Dr. Suresh Mohan Kumar, our research director, sir, please.
Thank you very much. Good morning and good evening to Dr. Saravanan as well. Ravi, I think Mainathan sir smiling and Dhanpal sir smiling, JSK sir smiling. You answered the Viva very well. So, for the first time. Actually, my question is open to both of you, and I congratulate both Dr. Saravanan and you for the enlightening session, and it was very informative, and as principal rightly said, this is the right topic at the right time with the right experts, and you definitely proved, I'm sure the thousands of watching this would have benefited from this informative session. So it's just an ideological question from me. See, I think Dr. Saravanan also mentioned this, there is a threshold, and you also mentioned there is a threshold clearly mentioned in the ICH guidelines. But more and more we dig the recent cases that we see, it is beyond that threshold limits. Again, the losartan, the case that he measured, and the ranitidine, that case that we saw, it has clearly passed the threshold limits. But the impurity seems to be very low at the base level. So first, what is the thought process on that? Again, there is a distinction between the manufacturer and the regulator. So the manufacturers don't want to decrease or more strict the threshold limits, because it is increases the cost burden on him. Whereas the regulator concerned about the safety issues, and he wanted to bring the threshold levels if more studies are done like that. So maybe I would like both of you answer very briefly and shed some lights.
Yeah. See, as someone who involved in this losartan as well as the valsartan process during my stay in Aurobindo, I can definitely say that one of the biggest problem or biggest thing is once it is commercialized after the R&D, there are a lot of changes usually happens in the process and the controls. So though we have a system to track and approve these ones, what happens is the characterized and listed impurities are the only one which is commonly focused during the routine manufacturing. So that's the problem happened in both losartan case and the valsartan case, for that matter, for every sartan case. So first we need to go back a step and see where from this NDMA or NEMA, this impurity sources are arrived. So that is started from intermediate and solvents which are used during that process, and from there. So the biggest challenge here is are the deviation happened here is on the process levels, those solvents had a certain impurity which led to the formation of this NDMA or NEMA. Even now for the metformin, it is being told. The other thing which we have to consider is the analytical knowledge or the analytical technique which was existed 10, 15 years back and today are vastly different. Because the development and scientific advancements give a lot of upper hand to current scenario. But when you implement that to the world products, obviously you get into the surprises like this. But at the end of the day, the patient has to be benefited from that. So the cost is not the matter here. For companies, obviously no company is doing service. The cost is going to be passed to the patient only. So the cost is not from the hesitant from the companies, but the cost will be hesitant from the providers. Basically, the US market, if you take the insurance providers, and those ones are the who will push back the cost to the patients. So this is something usually happens. So for example, one drug we can say the phenylpropanolamine, which was like 40, 50 years it is available in the market, and suddenly one day they came and said, oh, it is hepatotoxic, it is causing hepatic failures, you have to withdraw this drug. Yeah, that happens. This is called regulatory risk in the every business. But in general case, where we can control or how we can avoid this is one thing is companies has to start putting the controls beyond the final end process. So basically, usual controls are exerted on the final optimized process from the intermediate to the API. But this incident probably gives a better advantage or better understanding in the within the industry for the quality group to demand more controls, backwardly integrated controls. For example, the intermediates are the one which is a little bit less regulated in the API industry, or which is less seen as, okay, if it is a primary intermediate, then only I will focus quality development on that. If it is secondary or tertiary intermediate, they are like any chemicals which we buy from the Sigma. So those ones will now improve. Those focus will go up, and that will definitely improve the product quality going forward.
Thank you, Ravi sir. Now I request Dr. Saravanan, please add your point on this.
Not there. It's not there. Okay.
Okay. Anyway, Dr. Sarwan is not in online, I think. Anyway, as an organizing secretary, now I'd like to thank both the speakers, the resource persons, for your wonderful time and wonderful presentation. And before we enter into the vote of thanks, I'd like to pass some information to all the participants. The feedback link will be shared soon to your registered email ID. And the next will be your certificate. It will be generated after your feedback from our JSS Agar web system. And the next point will be within soon, I mean within 1 or 2 months, we'll be having a AICT sponsored one more impurity profiling e-webinar. Again, we are going to organize, we are trying to organize very soon. I request, anyway, all the registered participants will try to contact you through your email ID. And that particular AICT sponsored impurity profiling webinar session will be in a different way. You'll be having your e-presentation, e-poster presentation, as well as you'll be having a best presentation award too. So with this, I request now our department faculty, Dr. Babu, to proceed with a vote of thanks for this international webinar. Thank you.
Thank you, Dr. Jayaprakash. It's with the blessings of His Holiness. It's my pleasure to propose the vote of thanks on this international webinar. In the motto of the JSS Academy of Higher Education, that is learning never stops, we thank the JSS AHAR in motivating its faculty members to keep the faculty members engaged in such initiatives, both the faculties as well as the public and the students of the JSS AHAR, to keep them engaged in this COVID pandemic. So we would like to put our thanks to the JSS AHAR first. Also, we would like to thank our principal, Dr. S.P. Dhanapal, who was the motivational person in keeping engaged in bringing out all these sessions. It's not only with this particular one, we have been having various sessions with various departments, various specializations. So we thank you, sir, in motivating your faculties to be, in their legs in this COVID pandemic. We would also like to place our appreciation to both our speakers, Dr. Saravanan from Spinco Biotech and Dr. Ravi Jayapal from Citlagen, USA. We do understand, so one is presenting, giving you have taken your valuable time. One is the earlier part of your day, and another one is the later part of your day. So we appreciate and thank your enthusiasm in coming forward, in sharing and your expertise to the society in this pandemic, and to our students of JSSR. So we place our sincere thanks on behalf of JSS AHAR, JS College of Pharmacy, Ooty, to both of you speakers. We would also like to place our sincere thanks to our Vice Principal, Dr. Afzal Azam, our Administrative Officer, Mr. Baswalinga Devaru, our Research Director, Mr. Suresh Mohan Kumar, and our CIO, Dr. Ravindra. I think you, sir, you have been busy for the last 4 months, and we really appreciate your team, in putting these events very successfully reachable to all the people around the globe. And again, we also would like to place our sincere thanks to our internal technical faculty. I would like to place it in this particular term, Mr. Jayakumar and Mr. Gautam. And I think, they are also, along with his colleague at the JSS AHAR, Dr. Ravinder, I think, how difficult it is. They are sitting there for 3 months, and, all from engaging the JSSU online classes to the students, parallelly running all these sessions. So we would like to place our thanks to both these faculty members of the college. And we would also like to put up appreciation to the head of the Department of Departmental Pharmaceutical Analysis, Dr. Krishnaveni,
its faculty members, Dr. S.N. Mayanathan, Dr. J.S.K. Nagarajan, Dr. Jayaprakash, and Dr. Babu, in trying to bring out this one particular sessions. And we'd also like to thank all the university officials who have been part of this evening, though we have a late evening for us. And I think we like to place our all sincere thanks to all the university officials, the IIIC coordinator, Dr. Gautama Rajan, and the IIC members of these institutions. And last not but the least, we would like to place a sincere thanks to all the participants. I think, we really appreciate and we sincerely apologize because of, we do have the limitations in our analytical techniques. We do also have the limitations in these technologies. Sorry, we could limit only to the 100 participants, but then we could see the enormous enthusiasm shown by various participants at our YouTube link. And I think, we could, in the coming presentations, we would try to increase the numbers so that, various people from the globe can also participate in this. So we sincerely apologize, and we thank all the participants who have shown their enthusiasm. And we could also see from their feedback, much effective. So we would like to place this into our speakers, both the speakers. Many of the, nearly about 90% of the participants have appreciated that the topic which was being selected and the presenters who have presented on these particular topics were really appreciable. And we also thank on this behalf all the members who have directly, indirectly supported this evening session. Thank you, one and all. Have a great day.
Thank you.
Thank you. Congratulations to your team, your team.